What is subcloning? Subcloning is the joining of two or more separate segments of DNA to generate a hybrid DNA molecule which are capable of autonomous replication in a given host. There is two methods used in subcloning, which are TIA cloning and RE cloning.
First of all, we will be discussing TIA cloning. The T stand for thymine while the A stand for adenine. This subcloning method, which clones PCR amplified products into a suitable vector without full use restriction enzymes. Instead, this method was based on the ability of thymine and adenine bases on different DNA fragments to hybridize and ligate with each other at the presence of ligase.
TIA cloning was done by amplifying the product or insert using Taq DNA polymerase, which adds an adenine into the 3' end of the insert. The amplified inserts are then cloned into linear vectors that have 3 thymine overhangs. The diagram shows a simplified flow of TIA cloning. The process in green arrow is the manual method, which require restriction digest, while the process in blue arrow is a PCR-based method which does not require restriction digest. Regardless the method used, the aim is still to create a thymine-adenine overhang which allow the insert to be ligated with the vector. There are a few advantages of TIA cloning. Firstly, it allows cloning of DNA fragments that are not available in large amounts. Secondly, this method is also faster and simpler than traditional subcloning, as it does not involve restriction enzymes, except for the generation of insert. It also saves time and money because shorter primers can be used, as there is no need to add restriction sites when designing the primers. Last but not least, it can be used when there is no viable restriction site present. However, there is still limitation in TIA cloning. Firstly, directional cloning is not possible in this cloning method, hence there is a 50% chance that the gene can be cloned in a reverse direction. Secondly, the choices of vector is severely limited. RE stand for restriction enzymes or restriction endonucleases. This cloning method involves using restriction enzyme. Restriction enzymes cut DNA at specific recognition sites, restriction site. There are two types of cutters in restriction enzyme, which are the blunt end cutters, which cut target DNA at the same spot, and the sticky end cutters, which cut target DNA at different spots, creating 3 or 5 overhangs. The RE cloning is done as follow. The advantage of using RE cloning is directional cloning can be achieved more easily. Secondly, this method is also significantly cheaper compared to TIA cloning. However, there are also limitations. Firstly, there is a possibility of obtaining a vector-vector pair and a insert-insert pair. Secondly, the vector might religate. There is also sequence limitation, as it depends on the location of unique restriction site. Lastly, some of the restriction enzyme does not work well in commercially available buffer solution, which might result in low yield. To overcome this, using 2 restriction enzymes to prevent self-ligation, non-directional cloning, vector-vector pair and insert-insert pair. Secondly, treat vector with phosphatase to remove 5-phosphate group to prevent self-ligate.
Here are the comparison between TIA cloning and RE cloning. TIA cloning uses PCR method while RE cloning uses restriction enzyme. Secondly, TIA cloning is more simpler but RE cloning is more complicated. Thirdly, TIA cloning has limited vector choice while RE cloning has a more diverse vector choice. Next, TIA cloning can only achieve non-directional cloning while RE cloning can achieve directional cloning when 2 restriction enzyme was used. TIA cloning product has lower ligation efficiency while RE cloning has higher ligation efficiency. As a summary, subcloning is a technique used to join 2 separate DNA fragments. RE cloning is a cloning method which utilizes restriction enzyme in order to join DNA together. TIA cloning is a cloning method which utilizes Taq polymerase to create an overhang in order to join DNA together. Both cloning methods have various advantage and disadvantage which can be used at different condition to suit the need.
This is the reference used in this presentation. Thank you for spending your time watching this video. This presentation is brought to you by Sesha, Michelle, Cyanji, and Jason.